everyone, my name is TAI XIN and my metric number is S74400. Today I'm going to explain what I have done in Lab 2 of Biochemistry, which mainly about making dilutions, especially using the serial dilution. So first of all, we have a stock solution of 20 milligram per ml. Because this one, when I scan, it cut a bit of the edge, so I just rewrite the concentration here. So this is 20, and the final volume required is given, which is 50 ml. So the dilution factor is also given, which is 50. So based on this formula, dilution factor equals to final volume divided by the initial volume. So that we now we have the dilution factor, and we also have the final volume. So this is the final volume. So okay, this one I need to point at here. Okay, so this one, like this, we can sub these two value in the dilution factor formula, and we can get initial volume equals to 1 ml. So the solvent we need to add into the tubes is 49 ml, because 50 ml is the final volume, and the 1 ml is the initial volume, which means we take it from here, the stock solution. So 50 minus 1 ml, we get 49 ml. This is the solvent. Okay, so the solvent we already fill in the test tube, and now we transfer. I use a blue pen. I transfer 1 ml from the stock solution into here, 1 ml. And then 49 ml is the solvent, which we calculated just now is 49. So we fill in the 49 ml of the solvent in the test tube first, and we add 1 ml from the stock solution, and the total volume will come up to 50 ml, and then mix well. And then we take 1 ml again from here, the first test tube, from here into here, 1 ml. And then again we fill in the 49 ml of the solvent first, and then we have 50 ml, and then we mix well. And then again, this is the last step, we add in again the 1 ml from the second test tube. This is the second test tube to the third test tube. So this is, we also do the same thing, we add 49 ml of the solvent, and then the total volume will come up to 50 ml, and then we mix well. So the question asks, how much would you add into the test tubes? So obviously the answer is 49, because the answer is 49, because from the calculations we know that the 1 ml is the initial volume, which means we take from the stock solution, and this one is the solvent we need to add to fulfill the final volume of 50 ml. So the answer obviously is 49 milliliter, because the final volume is 50 ml. Okay, so now how would the tubes look like after serial dilution was done? Okay, so basically what we do just now, the transfer of the 1 ml solution is not as serial dilution. This is not as serial dilution. Serial dilution, which means this step from here to here is not as serial dilution. So the question asks, how would the tubes look like after serial dilution was done? So how would this tube, and this tube look like after serial dilution? So obviously the first tube will left 49 ml, because they transfer 1 ml from the first test tube to the second test tube. So the first test tube will left 49 ml, and the second test tube also the same, because it transfer 1 ml from the second test tube to the third test tube, so it becomes 49 ml again. But the things comes different when we moving to third test tube, which means the third test tube finally will have 50 ml, because this is the last step in the serial dilution, because we have no the fourth test tube again, no, so we just until 3. So we do not transfer 1 ml from here to here. No. So this one we will left 50 ml here, the only for the third test tube. So how about the final dilution? So just now we already said in the beginning that the dilution factor is 50. So the first test tube, which here, obviously we have 50, and then the second test tube already 50. The second test tube currently have 50, and then you need to multiply again by the 50. So we get 2,500. And then the third test tube now have 205,000. And then it need to multiply again with this 50, with this 50. So it multiply again and we get 125,000 dilution factor. So the last question is, what is the final concentration in each tube? Concentration. So we need to look through all the picture first, and we need to get the full image of this serial dilution, and we can get that first of all, we know that the stock solution concentration is 20. So each of the formula in here is the same. It starts with 20 because this is the initial concentration of the stock solution. The only difference is the dilution factor. These three are the dilution factor. So obviously in the first test tube the dilution factor is 50. So we divided 20, which is the initial concentration of the stock solution, divided by 50. The dilution factor we get 0.4 mg per ml. Mg per ml because we need to refer back to here. Because this one also state that mg per ml. So it should be the same unit for each of the. So for each test tube, the concentration should be using the same unit. And then this one, we're doing the same, just this one different. Will be a bit different. This, I use blue color. This one, different. It's green, blue. Okay, this one different. This one. So this one will be different. This the dilution factor will be different. Dilution factor. This is 50. This is 2,500. This is 125,000. And after we divided by the dilution factor, and the first test tube, the final concentration is 0.4 mg per ml. The second test tube is 0.008 mg per ml, and the third test tube is 0.00016 mg per ml. So these are my understandings based on the lab 2 practical that I have done that day. I hope you all can understand what I've explained today, and thanks a lot for watching my video.